some water, it's not going to sink as fast as the ribosome that you and I have. What do you think that ribosome in that chloroplast, how fast do you think it's going to sink? Because it's going to sink like a prokaryote. It's actually, the ribosome in there is the same as the ribosomes in the prokaryotes, in the bacteria. Which brings up a really interesting point about the next two organelles, the chloroplasts and the mitochondria. The idea for these two organelles is that they're actually bacterial invaders. Some million some odd years ago, there was a big bacteria, and it ate a little bacteria. But when it ate it, it didn't kill it, and it got something good out of it, like food. So it's like putting McDonald's into your kitchen. No, you don't have to go out for food anymore. It's right there. This big bacteria ate this little one, but it made food for it. So why would it want to kill it? So it kept it around. And it kept it around and kept it around until they became one and the same. This idea is called the endosymbiosis theory, and it was proposed by a lady named Lynn Margulis back in the 60s and 70s. Well published and kind of clarified by her at that time. And so we see with these that there's actually double membranes. There's a membrane on the outside, and there's some membrane-bound stuff on the inside. So they're pretty neat structures from that standpoint. So that's our chloroplast that we see in plants. And if we look down to our next organelle, we have the mitochondria. We see this in most eukaryotes. Both plants and animals will have mitochondria. And this one, if you look at, has the same kind of features. Circular DNA, has its own little ribosome running around in there. And this thing makes energy. And so the idea is this got eaten long, long, long ago. And it gave like, a boost, like a power boost to that cell. So what we see is that what we see with that one is that it will make us lots and lots of energy. It does that because if you look in there, you see lots of folds, folds on folds on folds, and you've got a little picture down here. It's actually a TEM image that they've gone in and put some color on for you. You can see these little networks back and forth and back and forth. What are those doing for us? If I have lots of folds, what does that do for me? Well, it increases my surface area without having to increase my volume. So I've increased my surface area for the machinery that will make energy for this cell. Lots and lots of energy. And a cell needs a lot of energy. Remember we said a cell is an open system that has to kind of look steady. It's got to bring stuff in, it's got to kind of manipulate it and use it, make new things, it's got to ship stuff out. That takes energy. I mean, if you're going to go out and walk down to the mailbox and get something, come in, open up that mail, put something in an envelope, walk it back out to the mailbox, that takes energy on your part. Well, that takes energy on the cell's part too. How are you going to feed that energy debt? Well, you're going to use food to make energy. And our mitochondria use food to make the energy currency for the cell. And a cell is going to use energy in the form, usually, of ATP. That's its most favored method. So the mitochondria takes food and takes oxygen and helps the cell to get lots and lots of ATP from that. A cell doesn't have to have mitochondria to get energy. If you look at something like the bacteria, they don't have organelles, but they're alive and they eat and they get ATP energy. But they just can't get as much necessarily as something with this mitochondrial structure. They have some mechanisms for getting energy, but they're not as good at it as these things that have the divisions of labor. If I don't have my mitochondria working very well in a cell, I can get energy. Usually on the scale of I can net about 2 energy units. If I throw for one glucose I can net 2 units of energy. I throw a mitochondria in there, for every one single molecule of glucose, I can net anywhere from 28 to 36 units of energy. Really, really much more efficient once we have that mitochondria in the cell. Now, there's a payoff, though. Think about anything that uses energy. Think about your car when you put fuel in it and then you run down the road. When you stop and you put your hand on the hood, what's it feel like? It's going to be hot because your engine's not efficient. It takes that energy and it loses energy as heat. Same thing happens for this cell. Same thing happens for this mitochondria. A lot of the heat that we actually produce comes from our mitochondria as they're utilizing our foods, and then they're making the energy, the ATP currency. They're losing heat because they're not 100% efficient. Nothing's 100% efficient. So they lose a lot of heat in that process. So that's kind of all of the membrane-bound organelles that we have running around in the cell. That doesn't mean that we're completely done with all the structures in there. We have a couple of other things that are pretty important. A cell is not just a blob of stuff. It's very ordered and structured. You're moving things around and transplanting things here and there. You're going to need something to do that. Anything that's doing anything in the cell is because of our good friend, the protein. So we have within the cell protein networks, proteins that form fibers together and basically make up the cytoskeleton of the cell. Very, very analogous to the skeleton that you have in your body. If you just had a skeleton, you'd probably have some problems. You need muscles overlying that. You need tendons to help link things together. The same thing is happening inside an individual single cell. You have the cytoskeleton that provides structure, allows the cell to move, allows movement of things, materials inside of cells that transport. It's this dynamic, interwoven process that occurs. So you have a lot of overlap. These are networks together. You need all of these to really have your cell function properly. There are a number of diseases, actually, that are errors in cytoskeletal elements of the cell. So we have three major types of filaments that make up the cytoskeleton, three major types of proteins. And again, things are not really all that complicated in science. It's not too bad. We have three filaments, and how do we distinguish them? Size. We say, oh, that one's the biggest one. Oh, that one's not the smallest, not the biggest. It's in between. Oh, and that's the littlest one. And that's how they're organized. And so what we have are, based on size, our first one we have here are the microfilaments. They're micro, so they're small. They're the smallest filament. And there's a little picture of some cells that have been stained for microfilaments that are all red up in the topmost part of the slide. All those red cells, they've been stained for microfilaments. And you can see those cells are packed full of them, just tons of microfilaments. It just glows like a Christmas tree. And this is kind of like the muscle of your cell. In fact, the microfilaments, if you think about your muscles and how they move, lots of microfilament elements are involved in that. It's actin. A lot of your actin is a microfilament. And so this is like the muscle of your cell, helping things to contract and move around. It's the smallest one. Our next one, not that, trickily named. It's not the smallest, but it's not the biggest, so it's the intermediate one. The intermediate filament is probably most analogous, actually, to your skeleton. It stays around the longest. It doesn't change that much, and it gives a kind of a solid framework for the other elements to come in and attach to. So it kind of forms a framework in the cell. And if you look at the second picture there on your slide, it's all green and pretty, and that's staining for your intermediate filaments. A lot of those are in there as well, and they're large, so they take up a lot of space. Now your largest filament is actually your microtubules. So it's a whole bunch of filaments kind of together, and they form these tubes, and they're really, really big. You don't have as much of them in the cell. That last picture is stained. The green is the actual microtubules, and you'll see there's just not as much. But these are the superhighway. Remember we talked about, going back to earlier in the lecture, we talked about having that transport vesicle come out of the rough ER, and it needed to go to the Golgi. Well, it wasn't just going to magically float itself down there. It needed direction. Well, a lot of times it hitches a ride on a microtubule. So they're kind of the superhighway where things get directed onto and around inside the cell. Also, if anything extends outside of the cell, usually it's a microtubule, which brings us to our next slide, which is about cell extensions, things that extend outside of the cell. So we have two major types of cell extensions, and these are made up of microtubules. in our eukaryotes. The first one are